Okay, so we're going to begin the final session of the day today, where we're going to really focus on the effect of early and really very early life exposures on development of late life health outcomes. So we heard today, this morning, from Gloria Dominguez, set the stage on just understanding the diversity in the microbiome in early life exposures and its contribution to early life exposure. So this afternoon, we're going to have two talks, one by Margaret Karagas from Dartmouth College, who will describe the epidemiology of early life exposures to environmental toxicants, the microbiome, and the human disease. And then after Margaret, we'll have a presentation by Marty, and I'll talk about that in a minute. So thank you. Okay with it.
Thank you so much. It's wonderful to be here. So I'm an epidemiologist, so I'm coming from a perspective of trying to find the environmental influences on child and lifelong health. So I'm excited to begin examining the microbiome. Okay, and I'm also thankful to my previous colleagues and speakers for setting up my talk, where I'll be talking about in utero and early life exposures as a vulnerable window and evidence on arsenic-induced immune-related effects, just as one example of an environmental contaminant. I'll be talking about our New Hampshire birth cohort study and the potential sources of arsenic exposure during this vulnerable window, emerging epidemiologic evidence on immune-related disease impacts, and preliminary evidence on the effects of arsenic on the microbiome. So we've already heard from Dr. Liu, arsenic is a known carcinogen. I was involved in the IARC monograph where we deemed arsenic a skin, bladder, and lung carcinogen. But we also heard that it's involved in a myriad of diseases, including lung disease, cardiovascular disease, diabetes, growth, neurodevelopment, infection, and mortality. And much of this data has come from places like Bangladesh, where very highly exposed populations to drinking water contamination, which is widespread throughout the world. 100 million people worldwide are estimated to be exposed to levels above the standard, which is 10 microgram per liter. So we also know that arsenic passes through the placenta. So we already heard from Dr. Liu about the complex metabolism of arsenic, arsenite reducing to arsenite, and then being monomethylated and dimethylated with acetinosyl methionine as the methyl donor. And then there's a form that's found in fish, arsenobetaine, which is unmetabolized and unchanged. And you can find that the correlation between maternal blood levels and cord blood levels are very highly correlated for each of these metabolites of inorganic arsenic. So there's also a really interesting natural experiment that comes from region 2, Chile, Antofagasta, where there were very high levels of drinking water contamination for just a limited period of time. So they could look at people who were exposed in utero and during early life to these very high levels that were orders of magnitude higher than the EPA standard and the WHO standard. And those people who were born during that time period had very high mortality rates from bronchiectasis, which is a sequelae of chronic infection, and other diseases like cancer. And so this is an experiment that let us know that early life exposure may be very important. And then this has been recapitulated in mouse experiments as well, and previous speakers have mentioned Mike Walkey's interesting work. And another point to note here, and we've been talking about sexual dimorphism, that in utero exposure did have a sexually dimorphic pattern in the mice. And then many places around the world with high levels of exposure have found increased risk of infection related to arsenic and alterations in immune response and function. And this includes Chile, Bangladesh, Mexico, Thailand, and experiments in mice that were done at Dartmouth where the mice exposed to arsenic had delayed clearance of the H1N1 virus and actually more severe infections and other immune perturbations. So we also heard from Dr. Liu that low levels of arsenic are prevalent in our water supply, and this includes the New Hampshire population, where arsenic was mined in New Hampshire until the American West opened up to mining in the late 1800s, so we were the mining state. The other unique part about our population is that we use private wells. So about half of us have private wells at our house that are unregulated, and about over 10% of them have arsenic above the MCL, or the maximum contaminant level, and this is largely attributed to the bedrock geology. So for most people, diet is the main source of exposure. And I was also on an EFSA report and a scientific opinion where it was written up that young children have 2 to 3 times higher arsenic intake than adults. And some of the foods that are high in arsenic include rice and rice-based products, as well as some seafood and algae. So there is some concern in the United States because we don't have any statutory limits right now for rice. The EU has set some standards and is specifically focusing on infants. So arsenic enters the rice grain through silica transporters. Our crop fields have arsenic from pesticide residuals. Arsenic was used as a pesticide prior to DDT. This is an image of a rice grain where you can see a little bit of red, if you can, entering the ovular vascular trace of the rice grain. Arsenic varies widely throughout the world in rice, but the United States has some of the highest concentrations of arsenic in the rice that we grow here. So to look at environmental impacts on children's health, we started a pregnancy cohort study. And to estimate exposure, we get a variety of exposures. We ask questionnaires, we collect samples, drinking water, urine, dietary histories, and we do this longitudinally. We call people up intervally, every few months. When we collect samples, we get their 3-day diet, we collect breast milk, and a variety of samples. So far, we have 1,500 mother-infant pairs. We're still recruiting, relatively good response rates, and we're targeting private well users. So in our first paper, we found that both drinking water levels of arsenic and rice consumption were related to maternal exposure to arsenic in their urine. We also found high arsenic levels in a brown rice syrup, and then a toddler formula using brown rice syrup had 6 times the EPA drinking water limit. And then more recently, we published that formula-fed babies had higher arsenic than breastfed babies, and I'll come back to that later. During mid-infancy, this is an IMPRESS paper where we've estimated that it takes only 3 servings of rice cereal to exceed formula mixed with the drinking water standard of arsenic. And then this is another IMPRESS paper where we do show in turn that babies who are fed rice have higher urinary arsenic levels, and this is in 1-year-olds who are eating a variety of rice products. And then we've also shown in NHANES that children who eat rice and rice products have higher urinary arsenic levels. And this is important because rice is a staple food, it's used in rituals, and in our study, 80% of infants were introduced to rice cereal in the first year of life, and at age 1, 55%, the majority were eating rice or rice products in the past 2 days. So what are the health impacts? Well, our focus has been on the immune system to begin with, because we know that the immune system develops early, and that if we perturb the immune system, that it can impact lifelong health and disease, as you heard. The other point to mention as an epidemiologist is that infections are the primary source of morbidity and mortality worldwide. Many of us do not study childhood infection. It is very common and why we take our children to the doctor. We've also heard that there are many number of immune disorders that are on the rise, including allergies and atopy, on the rise in the US and elsewhere that are likely due to environmental agents. So in our mechanistic and clinical studies, as I mentioned, we collect a variety of matrices to collect exposure repeatedly. We collect cord blood and we collect placenta. And then we collect a lot of diapers. We have many, many diapers, thousands in our freezers right now, if you want to talk about data collection. So we're interested in, we collect meconium, thanks to Linda. We have meconium. We have the first stool that that is collected during the hospital stay. Then we collect stool at 6 weeks of life, and then at 1 year, and then we're going on to 2 and 3 years, et cetera. And then in a subset, we're also collecting 4, 6, and 8 month samples. And our clinical measures, for our immune response measures, we measure infection, as I mentioned, allergy and atopy through these interval interviews. We have immune profile from their blood, vaccine response, and IgE that we're collecting at 1 year for bloods, and we review their pediatric records. And I should mention that we're also collecting data on growth and neurodevelopment and performing a 5-year exam and repeated surveys for other outcomes, but I'll only be focusing on immune outcomes today. So with respect to infections, in the first 4 months of life, we found that the number of types of infections that children were reporting or having been reported by their parents was related to the amount of urinary arsenic or in utero arsenic exposure, particularly infections requiring a doctor visit or a prescription medicine. And then we went on to look at associations with specific infections in the first year of life. And I'd like to point out that nearly all children were reported to have an infection during the first year of life, and about half of those were treated with a prescription medicine. This is highly prevalent in the United States. And the associations we found with arsenic exposure included respiratory infection, diarrhea, lower respiratory infection, and wheezing. And what's interesting is that these findings parallel what's been found in Bangladesh, at least for diarrhea and lower respiratory infection. So the magnitude of the association is weaker, but the kinds of things that we're finding do mirror those that are in more highly exposed populations. So our mechanistic data support these clinical outcome findings. This is work we're doing with Karine Dow at Stanford, where we see a decrease in the number of T cells in the cord blood. These are the naive activated T cells, which could just diminish vaccine response or increase risk of infection. There's a shift and there's an increase in the Th2 cells, which if they're activated could lead to atopy. And then we see a very strong correlation between the arsenite transporter aquaporin 9 gene expression in the placenta and an inflammatory marker IL-1 beta. And that's work with David Robbins at University of Miami. And then we also use DNA methylation to infer immune cell profiles, and this is work of Carmen Marsa and Devin Kessler, where they did a DNA methylation array on our cord bloods and found that the CD8 positive proportion cells were greater with arsenic exposure. So this is our model, and this was from a review article we published a couple of years ago with the building on the concept that the microbiome impacts immune development, and immune development impacts the microbiome. So this bidirectional relationship where we're trying to look at early as in utero exposure and the early neonatal period and look at the environmental drivers of that developing microbiome and how that will affect disease risk. And what I'm talking about is infection, allergy, atopy, obesity, and other outcomes. So as you've heard, there is a hypothesis that arsenic affects the gut microbiome. And it is interesting, given our next speaker, that antibiotics that arsenic was an antibiotic prior to penicillin. It was used to treat syphilis. It's also been used as a psoriasis medication historically, so it may have immunosuppressive properties. And then we heard from Dr. Liu about the mouse model, how arsenic perturbed the gut microbiome composition and observed differences in the metabolic profile. So on the other hand, there is the hypothesis that we've heard that the gut microbiome affects the metabolism and toxicity of arsenic. So we heard about Tom Vanderwill's model where he sees pre-systemic arsenic metabolism as a significant process. And so we know that colon is a reducing environment, and so that it may create these other toxic species, like MMA3 is very toxic. And then, as we mentioned, the thiolated species have unknown toxicity. So we're creating all these new metabolites that we have to begin to understand. So in some of our early work, we looked at, and you've heard about the cystic fibrosis children. This is a cohort of cystic fibrosis babies that was set up by Juliette Madon in our center, and where we found that breastfeeding and the gut microbiome associated with the respiratory microbiome, and the introduction of solid foods was associated with the gut microbiome. And then in a subsequent paper, we looked at breastfeeding that was associated with the CF exacerbations, which tend to be the Pseudomonas, as you heard, and also that breastfeeding was associated with increased intestinal diversity and less respiratory diversity. And then in these graphs shown above, you can see that an individual neonate's microbiome tracks over time, and you can see the increasing diversity over time. And we see this increasing diversity in our birth cohort study as well. So we know, and as you've heard, you know, the microbiome starts being somewhat less diverse at birth and then begins to become more diverse until adulthood. So one of the papers that we recently published just this past week is on delivery mode and breastfeeding, two of the important factors that we heard earlier from Maria Gloria. And this is in our Healthy New Hampshire birth cohort study, that both delivery mode and breastfeeding were associated with the infant gut microbiome at 6 weeks of age. So it's not only just at birth, but it extends out to 6 weeks. And what we found was that it was actually delivery type, which was the stronger factor that was related to the unifrac distance than feeding mode, even at 6 weeks of age, although both were associated. And in our model, since we're now beginning to acquire enough epidemiologic data, we were actually able to control for the other factor. So finally, in our models, we're going to begin to adjust for delivery mode when we're looking at breastfeeding, and breastfeeding when we're looking at delivery mode. And I think what's important and new about this study is we looked at mixed feeding as well as exclusive breastfeeding. So this is very common and may be able to inform what we tell women. So exclusive breastfeeding differed from mixed feeding, and mixed feeding and formula feeding did not differ as much as we might have hoped. So I think this is an important finding that we'll hear more about. So I just want to tie back into the environmental contaminants. Breast milk has high levels of certain organic contaminants, like the DDT metabolites, but it has very low levels of arsenic. And as I mentioned, breastfed infants had very low urinary arsenic levels. So when we start looking at infant exposure to arsenic at 6 weeks of age, while we collect the diapers, we also collect their urine. So we collect both. And what we found was that among formula-fed infants, infant urinary arsenic concentrations were related to the gut microbiome at 6 weeks of age. So it differed by whether they were breastfed or formula-fed, and these are very preliminary results. But when we look at in utero exposure, there's a difference by delivery type, so by vaginal versus C-section. So maternal gestational urinary arsenic was not overall associated, but differed by delivery type. And the interaction is statistically significant, but again, these are preliminary analyses. So just in summary, we have epidemiologic data of an increased risk of infection. We also have new data of an increased risk of wheeze. And these findings are supported by our mechanistic data on lymphocyte profiles, epigenetics, and placental gene expression changes, and they, in many ways, parallel what's found in Bangladesh. We found that breastfeeding was associated with lower arsenic exposure and a distinct infant gut microbiome. and that we have preliminary data among formula-fed infants that the infant arsenic exposure relates to the gut microbiome, and maternal arsenic exposure and infant gut microbiome relationship is modified by delivery type. So I have many, many people to thank. This is a very large group effort led by Juliette Medan, who's a neonatologist, Annie Hohen, who's a bioinformaticist and an infectious disease epidemiologist, our student Sarah Lundgren, a former postdoc Sheree Farzan, Kathy Cottingham, who does a lot of the exposure assessment, David Kessler is a former postdoc, Hongzi Lee at University of Pennsylvania, Mitch Sogan at the Marine Biologic Lab, and Hillary Morrison, who've been doing our microbiome analyses. Thank you.
A few minutes. Are there any questions?
Hi, Isabel Walls. I work for USDA. So I noticed that you did have salmonella bacteria on your table, but you didn't mention an increase in foodborne diseases, and I wonder if that was also correlated in the infants.
You're asking if the infants had foodborne diseases?
If they had an increase in, you did show salmonella, but on your summary slide, you only said respiratory infections. Is it also foodborne infections?
Yeah, we don't know. Yeah, we didn't.
But you are looking for foodborne bacterial pathogens in the diapers.
Right. We're mainly looking at patterns at this point. You know, we're just beginning to explore the patterns. And one of the, we're looking at composition right now is what we're doing. This is 16S, and we're just beginning to endeavor into this. But that's a great question. We're just starting to scratch the surface of this. We're trying to establish a methodology, some epidemiologic principles, you know, some statistical principles and bioinformatical principles, and looking at methods, as we've talked earlier about, like, how do we look at these data? Should we be looking at networking methods? That's something Annie Hohen's exploring. So we're just beginning to be able to try to make some inferences. I mentioned that we're also, through a supplement, going to be looking at metabolomics, and then we hope to eventually look at metagenomics. And once we get more specific, I think we'll be able to address questions like the one that you're asking. So
and also, are you looking at socioeconomic status of the mothers and how that might relate to whether arsenic is in their diet?
We do look at socioeconomic status, and you know, it is not related to the arsenic exposure, but we do adjust for it. So certainly it's not related to the drinking water exposure, but it is a factor that we're interested in. And we're also, I think you bring up a really good point, we're interested in how the socioeconomic status or the social environment might also interact between contaminant exposure and the outcome of the child. So that's a very good point to bring up. Thank you.
Jermaine and then Gloria. Go ahead.
Hi.
Jermaine Buck-Lewis, NACHD. Thank you. That was a very nice talk. I was wondering if you have had the chance to look at infant sex related to all the vulnerability discussion today.
Yeah, that's a really great point, and one that I am taking home with me. And,
Oh, okay.
Yeah. So yeah, very much a great point.
Thank you for a nice talk. Can you expand a little more on the point about arsenic and delivery mode?
You mean about maternal in utero exposure, and you mean why that might be the case? Well, since we've already learned from you that delivery mode is very important, and we see that in our study as well, that the only thing that it's speculative at this point, what might be happening is just the impacts if the composition of the microbiome is different by delivery type, then the impact on that composition could therefore be different. So I think what we're beginning to learn is that we need to stratify and look at our data based on things like delivery mode and feeding mode, and that the impacts may differ based on those important factors. And in this case, we did see that. So at this point,
So you cannot say that arsenic was different in kids delivered by C-section or vaginally. It's all linked to feeding mode? Is that it?
no, what we're seeing is the in utero arsenic exposure's impact on the infant gut microbiome differs by their delivery mode. So it is what you said. That is what we observe.
Uh-huh. It's hard to imagine a mechanism, right? Do you have a hypothesis?
This is
So
preliminary.
what that suggests is that, so is it higher in vaginal birth than in C-section?
The association isn't, at this point, in this preliminary analysis, stronger in one versus the other. But what it's saying is if the composition is different by delivery mode, if the infant's gut microbiome is different, then the impact of environmental factors could be different because the composition is different. That is one hypothesis, for example. So I think this story is yet to be unfolded, but I just wanted to show an example of what we're beginning to look at in our data. We want to look at air pollution and all kinds of other stuff. So we're just starting.
Thank you.
So maybe you haven't done this yet. I was going to ask you about indoor air pollution, because in New Hampshire you have lots of people who are using wood smoke indoors, and we know that that may suppress the immune system as well and increase respiratory infections. So I was just wondering if you've begun to look at indoor air pollution.
Yes, that's actually one of my favorite topics. And you funded a K to look at this. Abby Fleisch at Harvard is looking at indoor air and actually has monitors in some of our pregnant women's homes. So we'll be able to validate some of the questions we began asking at the beginning of the cohort about whether people have wood stoves and how frequently they use them, et cetera. But it is true, just like private wells, probably, the vast majority of us use a wood stove because it's so cold where we live. So that is a very important question. And they vary by the type of wood stove. Some people have very old ones that are very leaky, and other people have tighter ones. And I think this is a very important area that we must understand.
Thank you.
Thank you.
It's very interesting.